Okay, hi everyone. And as this picture may imply, my talk is not going to be entirely like on microbial communities. I'm going to talk about some general models to test a couple of hypotheses on formation and evolution of species' range limits. Um, uh, These two hypotheses, one of them is generally known as genetic swamping hypothesis, basically says that if we have an environment, let's say that this environment is imposing a gradient on a fitness-related trait shown by a color gradient here, and let's say that these species are going to get adapted to this environment and then move their and expand their range. So the species that migrate randomly to the edge, they are pre-adapted to the core of the population. And once they move to the edge, and since the density at the core of the population is much higher than the edge, so they create a sort of predominantly core to edge movement, and then they create a sort of gene flow from the core to edge, which is going to be maladaptive at the edge, because these populations are pre-adapted to the center. And this maladaptation that they create at the edge is supposed to create, it's hypothesized that it's going to create range limits if the environment gradient is sufficiently steep. The other hypothesis goes back to Darwin and still under debate that interspecific competition is going to create range limits. Okay. So there have been a sequence of PDE models trying to test this hypothesis, starting from the work of Kirkpatrick and Barton. Is a highly influential work. They developed a single species model to test the genetic swamping hypothesis. And then that was extended by Case and Taper to a community of the species to test the competition hypothesis. We have been building on these models to relax some of their assumptions on the genetic evolution of genetic variants and also the dispersal types. I'm going to talk about these results that we have reviewed. So the model starts from this basic equation for each of the species in a community of the n species. So let's say that each individual is represented by its phenotype, which is denoted by P, is a continuous random variable, and its distribution is going to be, its distribution is denoted by that function phi at each point x in the space, each instance of time t. We assume that it's going to be normally distributed. It's a strong assumption, but it's going to be useful when we are deriving our mean field models. And then we assume that the population density of the phenotype P, which is the density of the entire population denoted by n, times the frequency of phenotype P given by phi, the changes in that density of phenotypes over a short time tau is going to be dictated by three components. First component is a diffusive movement, which is going to model the random dispersal of the individuals. Second component is due to intrinsic growth rate of the population. And third component is due to mutational changes. Okay. Let me show you the ingredients, which are relatively classic. So the intrinsic growth rate comes from the logistic growth. We can have Ali effect, and then maximum growth rate of the population, carrying capacity, competition shown by this kernel. Competition is going to be phenotype dependent, and then natural selection is going to act and penalize the phenotypes which are different from the optimum phenotype that is imposed by the environment. And S is the strength of the natural selection. Competition is modeled by the overlap formula between the resource utilization function of the species, each individual, but I call this phenotype utilization curve because we identify resource access by phenotype access. So the overlap between these resource utilization is going to give us the strengths of the computation. And mutation is modeled by this formula. We have a reduction in the frequency of the phenotype P because of the mutation from P to other phenotypes, and also an increase in the frequency of phenotype P because of the mutations from other phenotypes to that phenotype. And these mutations are going to be dependent on the distance between the P and other, like the distance between the phenotypes. We substitute these ingredients in that basic or like master equation that I showed you. Then we derive the mean field models by integrating, finding the first moment, second moment. And then we find these equations for the evolution of the population density, trait mean, and trait variance for each of the species inside the community. These nonlinear terms, G, H, and W, have these forms, and then those other terms inside that have these forms. So the equations are large. We have to solve them numerically. For that, we need to find reasonable parameter values. Since the model is abstract, we have to be careful about that. Fortunately, the parameters have biophysical meanings. We could spend a couple of months to find some reasonable range of values, and then we have to be careful also for choices of units. And this is typical range dynamics. You will see when two species are going to expand their range, initialized with this orange curve and green curve. They establish themselves, they expand through traveling waves. Once they meet each other, they start competing, and then competition is going to halt their range expansion. So they reach to an equilibrium at which both of them have limited range. And those equilibrium curves of population density, I'm showing that with green and blue. Here, I show the equilibrium curves of trait mean. And you see that over the region of sympathy where they coexist together, they show character displacement, which is a very well-known feature in coexisting species. So what mechanism is creating these range limits is important to know. What happens is that once they meet each other, there's going to be a competition between them. Competition is going to induce character displacement, and it's going to decrease the density. Once the density goes down, that asymmetric maladaptive gene flow from the core to edge is going to get intensified. Once it gets intensified, it further decreases the population density and increases more, intensifies also character displacement. And this process is going to finally reach to an equilibrium at which this maladaptive gene flow is going to destabilize the balance between selection and competition and create those range limits. And in order to see that this gene flow is really contributing to the formation of the borders, here I make the environmental gradient to be shallow. Once the environmental gradient is shallow, we see that this region of sympathy is going to expand to the entire habitat. We literally don't see any range limits. I should have said that I consider a linear environment in these simulations shown with this black line. And you see the species are showing character displacement over the entire habitat. So gene flow is really maladaptive gene flow is really contributing to that. But the fact is that these simulations that I was showing you was for identical, like competitively equal species, which we never have. All the time when we have a species, one of them is going to be stronger than the other one. So as long as the environment is linear, one of them is the stronger one is going to push the weaker one once they form their limits, and then toward the habitat boundary. It can result either in exclusion or a marginal coexistence, depending on the difference between the strengths of the two species. So why do we get this marginal coexistence? Here, when the difference between the two species is small, is because here the weaker species has lost its core. As a result, it's not suffering much of like core to age maladaptive gene flow. And that gives the species a sort of advantage that compensates for the disadvantage it has because of competition weakness. We identified a sort of environmental nonlinearity that can stabilize these competitively formed range limits. So if environment is not linear, but it has some sharp changes where the steepness of the environment changes sharply. These ranging, or which I call it knees, or we have some step-like changes in the environment, which we can see like ecotons in ecology. It looks like that. Then that moving boundary between the two species, once it reaches to that knee, it's going to get stabilized there as an equilibrium. Why does that happen? It's because the weaker species now is adapted to the shallow gradient, stronger one to the steep gradient. So stronger one is suffering more of maladaptive gene flow. This one is not. As a result, this one now has more advantage over the knee. And that is the mechanism that stabilizes the range limit. And it happens that this stable equilibrium is pretty strong against perturbations in the environment. So in this simulations, I just simulated climate warming perturbation. I just shift the curve of environmental optimum trade upward to say that it's just like a climate warming. And it happens that initially the stronger one gets some advantage, pushes the boundary toward the weaker one. But after that, after a while, the weaker one gets adapted to the newer environment and then pushes the range back. So this range stabilization can be stable, can be robust against environmental changes, and that gives us a message that places in the nature where we see sharp changes in the gradient are going to be less sensitive to environmental changes. So far, the mechanisms I have been talking about relies on the fact that there is a maladaptive gene flow, which is because of the assumption that the dispersal is random. But for many species, dispersal is not random. They disperse adaptively based on the information they collect from the environment. One of those mechanisms is known as matching habitat choice, in which individuals just assess the environment, they move to a location that best matches their phenotype. Let's say that this is an environmental gradient. The phenotype of each individual is shown by the colors. After matching habitat choice, if it is done perfectly, they find their best matching location and just they move there. Okay. There have been evidence for many species that they do that, for grasshoppers, for example, and also for microbial organisms. In this experiment, they tested this dispersal behavior on ciliates, which show genetic variation in performance over a thermal gradient. So they have 3 patches. They inoculated the species on the central patch, and then they let the species move to the other patches. These patches are maintained at 2 different temperatures, one of them at 23, which is the core of the thermal niche of the species, as well as 35 degrees, which is the upper bound of the thermal niche of the species. And then they see that those individuals who disperse adaptively to the upper range limits, they have higher fitness. And then they test to make sure that this behavior is not because of selection, it's not because of phenotypic plasticity, it's not because of drift, it is just because of this habitat selection. So this matching habitat choice has many serious eco-evolutionary effects, and there are many concepts related to that. So I just wanted to point out a few of them, that evolution of matching habitat choice depends on selection, but operation of matching habitat choice is independent of selection. And operation of matching habitat choice is mostly related, because it's a performance-based adaptive dispersal, it mostly depends on the niche breadth of the individuals, how sensitive they are to their environment. And adaptive dispersal can facilitate rapid adaptation, and more importantly, it is going to create adaptive gene flow instead of maladaptive gene flow. So gene flow to the boundary now is going to be adaptive under this dispersal strategy. Now this raises the question that can competition still create range limits if the dispersal is adaptive and gene flow is adaptive? because I just show you that maladaptive gene flow was contributing to the formation of range limits. Now, if the gene flow is adaptive, do we still get range limits or not? Okay, so we try to model that. Since the framework of the model is a reaction diffusion framework, the easiest way to model such a thing is by just adding an advection term to that master equation that we have. Let's say that is going to be just the advection term. It is analogous to the drift diffusion equations that we have in physics. When particles are moving, for example, in a fluid, this A is going to be mobility of those particles, which I call them dispersal propensity here. And this is going to be a force which is going to direct the movement of the particles, which can come from the gradient of a scalar potential energy, which I define here as a phenotypic potential energy. The gradient of that induces a force, which I call it the phenotypic dispersal force. Then we just need to define a good potential energy. And I define that potential energy to be just like P minus Q. Q is the optimum trade value over 2V, which is the niche breadth. Okay, that potential energy gives us a measure of how poorly this individual, this phenotype P is performing where the optimum is Q. It definitely depends on the niche breadth of that individual, but it is independent of selection. So we get the gradient. It induces a dispersal force, which is going to direct the movement of the phenotype along the gradient in the optimum value of the environment, depending on the niche breadth and the mismatch between the phenotype and the environment. And we derive the mean field model for this case. It appears that this dispersal, adaptive dispersal, is going to affect the population density, mean value of the trait, and variance of the trait in different ways. Each of these terms are interpretable, and then they can create also a mechanistic insight about the operation of the adaptive dispersal. I'm not going to go into the details, but we see that is probably surprising that not only adaptive dispersal is going to prevent the formation of range limits by competition, but also it's going to make it even better. Range limits are sharper, and this region of sympathy is just like smaller. Okay, so that even though we have adaptive gene flow, so it means that in this scenario there must be some other mechanism working on. It's not maladaptive gene flow that is functioning. Okay, so and this is the curves of the trait mean. We see that when dispersal is only random, I just showed you that we have a character displacement over the region of sympathy. When dispersal is adaptive, this character displacement is substantially reduced. So these species can coexist without showing character displacement, but over a shorter region. And of course, adaptive dispersal makes the adaptation much faster than random dispersal. We did some calculations to make sure that this adaptive dispersal is really creating adaptive gene flow by measuring the effects of different dispersal components on rate of change of the trait mean, as well as rate of change of the population density. I'm not going to go into the details, but let me just tell you what happens in this cartoon. Okay, so let's say that I have two species. Individuals are shown by circles and diamonds. The environmental gradient is that correct gradient. Once they move toward each other, they overlap. Competition is also inducing some like character displacement. You see that the individuals over the region of overlap, they are showing character displacement. The color of the individuals are very different from each other. But this is the case when they are dispersing randomly. Now what would happen is that since these individuals are far away from their phenotype is far away from the optimum, matching habitat choice starts to act and push them back toward the core. because this individual now has to go back because it's not feeling well there. Same as for this individual. And this process is appearing is occurring dynamically, not the way that I'm showing it like that. So simultaneously they are going. As a result, eventually what we see that is going to be a reduced region of sympathy and reduced level of character displacements, because all those individuals who were at the interface and were not feeling well because of the induced character displacement, they move back. So the mechanism that is creating range limits in this case is backward edge to core movements that is induced by adaptive dispersal. We tested these differences between random dispersal, adaptive dispersal based on the different levels of environmental gradient, different level of selection strengths, as well as different level of specializations of the individuals, means that different levels of the niche breadth of each individuals. Since I don't have time, let me just let you read this part, and then I can take the questions. Thank you so much.
Questions from trainees? General questions?
Hi, thank you. Very interesting talk. I was wondering in the first case where there was maladaptive dispersal, in that case you saw divergence of the traits in the area of sympatry, but then there was a sort of extra divergence of the trait right outside of the area of sympatry.
There was? I don't think. Yeah?
Yeah.
Oh, you mean this?
Yes.
This is outside the range of this species. So the model is just like solving the equations for the entire habitat. But over here, the population density is 0. So that part of the curve has no meaning.
Do you have an intuition for what would happen if you considered mutualistic interactions? May gradients have stronger effects on setting range limits?
Yeah, good. Yeah, I mean, not much of intuition, but one of the posters I remember, I don't know if, but we are gonna see still. It seems that we are gonna see range limits, but probably it's gonna be like narrower, or like it might have some other features. Yeah, but I don't have much of intuition what's gonna happen. So that this model was trying to test a competition hypothesis. So.
Again, dragging it into my world, if you, instead of thinking about recombination, thought about mechanisms like horizontal transmission of plasmids, could that generate similar patterns of
horizontal gene transfer? You want talking about that? I mean, this model, that doesn't really model the reproduction. Yeah. The horizontal gene transfer, I would say, it is gonna be a source of, I would consider it as sort of like genetic variant, generation of genetic variation. Mm-hmm. I doubt it's going to change much, because the main source of genetic variation when we have steep gradients is gene flow. Probably that genetic variation created by gene transfer is not going to contribute much when it's not going to have a dominant effect when main effect is coming from gene flow. Because I didn't see any like big effects from the mutation as well, because mutation was present and was creating some genetic variation, but it didn't have much of effect.
Interesting.
Yeah.